Hello everyone. Thank you for joining my session. I am Arinjita Bhattacharya. I Can you hear me well?
Yes, we can hear you.
Okay, thank you so much. So I am Arinjita Bhattacharya from Merck, and I'm an associate principal scientist there. And okay, sorry, I'm starting my video. Can you see my video?
Yes.
Okay, thank you. So I'm Anjita Bhattacharya from Merck. So I'm the chair for this session. So chair and organizer for this session, and my session's name is Innovations and Trends in Oncology Clinical Trials. We will start with, unfortunately, on Dr. Anona Roy could not join today because of unavoidable circumstances. We will start with Tian He. He is a senior scientist in biostatistics from Biostatistics and Research Decision Sciences Early Oncology at Merck. She obtained a PhD in biostatistics from Indiana University, Indianapolis in 2022. Her research interest includes master protocol, dose finding, and Bayesian methodology. Uh, uh, so let's welcome Tian. And Tian, you have, since I am the second speaker in this session, so you are welcome to use at least 20 minutes from now. So at least like 17 minutes, including questions. So please go ahead.
Okay, yeah, thank you. Let me turn my camera on. It, it seems like something wrong with my camera. So yeah, maybe it seems like it does not allow me to share my like but let me share my screen first. Can everyone see my screen? Okay.
Yes.
Yeah, thank you. So yeah, thank you, and thanks for the opportunity to share our recent work about the backfilling strategies for point design in early oncology trial with late onset outcome and the dose optimization. This work was calibrated with Xiaohan Chi, a PhD student from MD Anderson, Ying Jieqiu from UT Southwestern, and my colleagues Yixin Ren, Hen Zhou, and Fang Liu. So my today's talk gonna starting from a brief background, followed by the method, simulation studies, and a summary. So firstly, let's starting from the background. We know that in most current phase 1 clinical trials, we firstly find like the MTD or like the maximum tolerated dose that has a targeted toxicity to the probability of 5 or most close to that probability. And then based on totality data and decide like which dose level to move forward. And the point design is one of the most popular way we use recently, which calculates the DLT rate at the current dose level and then compare the DLT rate with the lambda E and the lambda D here, these two boundaries, and then decide whether to escalate, de-escalate, or stay at the current dose level. And when using the Boeing design to do the dose selection, like after a certain number of patients enrolled the trial, we need to pause for a while, like the DLT window, to wait all patients have their have their events or like finish their DLT window, and then decide like whether to which dose level to move forward, and we need to assign the patient to which dose level. And with this backfill, but when patients are coming at this time, they can only wait until the trial open again. So in order to get some more information with, especially with those patients, we can consider backfill those patients to the lower dose level or the clear dose level. And in this case, with the similar trial duration, we could have more patients enrolled, and additionally can have some more information collected. And in addition to the backfilling, the FDA in 2022 also proposed a Project Optimus, which suggested to reform the dose optimization and the dose selection paradigm on oncology drug development. We can see that the left plot here shows our traditional way that we assume the efficacy and the toxicity have some mono relationship, and when we find the MTD, we should at the same time find the OBD with the highest efficacy rate. But when we see the plot on the right here, we can see that even if the dose level 3 and dose level 4, they have almost like really similar efficacy rate, but the toxicity rate in dose level 4 is higher than the dose level 3. And in this case, of course, we will prefer to choose the lower dose level with similar efficacy rate, but much more safe. So, and now we can call this lower dose level as OBD, the optimal biological dose. And where's my mouse? And we believe this is the dose level we will prefer to choose. So in order to like jointly consider toxicity and efficacy and also including the backfilling in the dose optimization, we propose our new method. And firstly, to use the toxicity and the efficacy information at the same time, we can use the utility here. This utility was proposed in Boeing 1.2 design before, which considers the utility as a function of different, like for use here, like U1, U2, U3, and U4, as the 4 different categories, where the U1 is at the score 100, which is no toxicity and efficacious. Is of course the best scenario we want to see, so it has the highest score. And the U2 here is no toxicity and no efficacy. U3 is toxicity and efficacy, and the U4 is too toxic and no efficacy, which like we never want to see. So its score is just 0. So based on these different scores and also the parameters theta here can be estimated based on the Bayesian design. So with this information, we can calculate the mean utility for each dose. And so when we do backfilling we know that usually there are multiple lower doses when compared to the current dose level. So we can only open the doses for backfilling when they are safe enough and they have certain number of responders, or like when the sample size is too small to observe responders. However, when we observe too many DLTs with a higher DLT rate when compared to the boundary here, those dose levels gonna be stopped for backfilling. And of course, there should be some scenario that there are multiple doses open for backfilling. And in that case, the patient is gonna be assigned to the optimal dose with the highest utility, where the utility is just will be calculated here. And in addition to the backfilling during the dose escalation, we will also consider the extended backfilling. And when we're doing the extended backfilling, we does not need like the DLT window, like the dose finding part here, and this can. And with extended backfilling, we can get some additional information when make final decision. But the extended backfilling is not open for all open dose either. We need to firstly find an admissible set, which similar to the backfilling during dose escalation or dose finding, which is safe, with the dose level lower or equal to the MTD, and also have certain number of responders or like the sample size too small. After that, in the admissible set, we will select two doses with the highest 2 mean utility and allow those two dose levels for the extended backfilling. And in addition to that, we know usually especially the efficacy endpoints, we need a certain number of months for the follow-up to observe the response or observe the endpoints. So this follow-up may take maybe like 6 months or even longer. But in the dose finding study, it is almost impossible to wait until all enrolled patients to finish their efficacy window. Otherwise, like this trial duration would be really, really long. That's like everyone does not want to see. So in order to accelerate the trial, we can consider the estimate or predict the outcomes based on the current observed information instead of wait all patients have their results. And this design was also applied in the tight point design. However, like it does not mean that since we can do this prediction, we do not need wait. We does not need any pause during the study, during the trial process. Actually, we also consider this two rules for trial suspension and allow patients for backfilling at that time. The first rule is that the trial will be paused for accrual if there are less than 51 patients have their toxicity, or if there are less than 31 patients have their efficacy outcome observed at the current dose. And the second rule here is that the trial could be suspended if the trial recommends the dose escalation and the shortest follow-up time for the pending patients at the current dose is less than 25 of the DLT assessment window. But it does not mean like when accrual is suspended, we can like the new patient cannot get treatment. Those patients can get treatment to the back, like they can backfill to the lower dose level that like I just introduced before. So here is a brief plot of our trial illustration. It is really straightforward that the left part is the dose finding with the backfilling, and the right part is the extended backfilling. And we can see that only two doses are open for the extended backfilling. And in order to have like better comparison, like for the dose levels, enroll more patients at the at the dose finding, they would enroll less patients in the extended backfilling. Now let's move to our simulation studies. In the simulation studies, we assume the scenario with 5 different dose levels and the sample size at dose finding at 30. The cohort size at 3, and for each dose level, the sample size at dose finding is limited at 12, and the backfilling part is 12 as well. We assume the DLT window at 2 months, efficacy window at 3 months, and the patient's accrual rate at 2 patients per month. We also compare our proposed design with 3 other existing designs, including backfilling point design, backfilling point ET, tight point ET. Well, the backfilling point design incorporated the backfilling into the point design, and the backfilling point ET consider the backfilling point with the efficacy and the toxicity outcomes. The tight point ET is the late onset point design with efficacy and the toxicity outcome. So here is the results of our simulation study. we consider four different scenarios here, and the OBD is cycled in green. We can see that in the bar plot here, the probability of correct selection of the OBD in our proposed design and the BF1 design are usually higher than other two designs. But when we check the study duration, it's straightforward that the BF1 design always have the highest, like the longest trial duration. And as for the sample size, this plot shows the sample size uh, and the tight bond design usually have the lowest sample size, followed by the BF bond ET. But when we check the percentage of patients that overdose in the first two scenarios, the BF1 ET always has a highest, or like the much higher percentage of overdose when compared to other designs, whereas the scenario 3 and 4 does not have the overdose. So as a brief summary, our proposed design can achieve the goals of those finding and those optimization in a seamless trial framework, and it can efficiently enroll and assign patients with the backfilling and the late onset imputation. It's really transparent and easy to implement. As for the potential extensions, in the future, when we backfill to patients to the lower dose level, it's enough just to assign them to the dose with the highest mean utility. We may can consider the adaptive randomization to allow some probability to be randomized to different doses. And additionally, because currently when we enroll more patients, we use a fixed number of like the cohort size, which is 3 here in our simulation. But in the future, we may can also consider to use a adaptive cohort size. I think that's all I have today. Thank you so much.
We have 2 minutes for questions. Do you have any questions for Tian? Thank you, Tian, for the wonderful talk.
Thank you. Can we take silence as no?
So did you apply this to any data set inside Mark or like anything that you?
Actually, no. One reason is like because for this kind of design, we need some it like when we enroll patients to different dose level, it high depending on the current like current status. So unfortunately, because most trials we we have our design at the very beginning, so we are unable to use it. But fortunately, we can use some simulation studies for it.
Okay, okay, great.
Yeah, thank you.
Okay, so since the next speaker is me, let me start. And thank you, Tian, again for the wonderful talk. So let me share my screen. Can you see my screen?
Not yet. Oh, yeah, I can see it now.
Okay, thank you, Tian. Let me do this. So hello everyone. So I'm Aranjita and I will be talking on this. And let me give you an overview of how I started my journey in the statistics world. So I started from India, and there I did my master's and bachelor's in statistics. I came for an internship here at University of Michigan, where I worked on a data set that is very close to my heart because it was about South Asian immigrants in the US, and which is known as the masala data. And it was really interesting project to work at University of Michigan Big Data Summer Institute. And after that, in 2016 I joined University of Louisville for a PhD, and there I worked on Bayesian shrinkage priors and clinical trials. After that, I joined Janssen for a summer internship there, and I worked on a Bayesian logistic regression models to do dose escalation similar to Bayesian and other designs, but Bayesian logistic regression were primarily used during that time. And then in 2020, I joined Mark, and since then I've been working at Mark, and I am currently an associate principal scientist at the Biostatistics and Research Decision Sciences. So that's my journey since um, from bachelor's to this stage. So I have been involved in many organizations here in the US in the statistics field, ASIC, Caucus for Women in Statistics. And I have been presenting at IDWSDS since last 3 years, and it has been an interesting journey. So I'm involved in NR, IISA, and if you are joining the Women in Data Science and Statistics conference in Cincinnati. I will be presenting there my talk, and also I will be hosting a session on mental health and hosting a panel session on mental health and um, invisible disability. So please join me there, and it will be great to see you there. So most of the therapeutic areas that I work on are oncology, vaccines, infectious diseases, and cardiometabolic diseases. So Mark has a lot of drugs in this area. Januvia is for diabetes, and then if you remember, we had taken MMR in our childhood, and also some of us has taken right now. So that is developed by Mark, and then recent Keytruda is one of the biggest blockbuster drug that Mark has developed. So it is the industry's most uh, largest immuno-oncology uh, portfolio that we have, and we received emergency use authorization from FDA and other agencies for molnupiravir, which was COVID-19 drug, and BCG is one of another vaccine that we have developed. So it's goes a long way, and I have worked on some of the infectious disease and cardiometabolic diseases. So let's start with early clinical development statistics, my department. And these are some of the therapeutic areas that I'm working on, which is HIV, oncology, and cardiometabolic disease, pain medicine, and COVID-19. So most of the trials that I work on are first in human studies, multiple ascending dose studies, bioequivalence, drug-drug interaction, formulation, and renal versus healthy population, QTC, medical devices, and other like actigraphy and other type of experimental medicine designs. So these are mostly held in healthy participants. So these are some of the studies that I work on. Let me see how much time I have. The slide is not moving. Okay, so the responsibilities that I have as a principal scientist here, and my life as a biostatistician is pharma is working in team with clinical scientists, data management, and programmers, authoring a statistical analysis plan for the protocol for a new intervention, conducting meetings with the team, designing studies. Mostly their parallel and crossover studies, attending drug-related meetings, then power and sample size calculations, preparing mock-ups for TFLs, and working with programmers to analyze the data. Finally, summarizing it in clinical study reports. And there are also job-related trainings, professional activities, and research activities. And Mark has a pretty good work-life balance, and I also enjoy that work-life balance. I travel a lot and with my husband, and it has been a great experience traveling all around the US. I have been to 40 states till now, and it's been a nice experience. So these are some of the studies. So let's come to pharmacokinetics and pharmacodynamics. So pharmacokinetics is what body does to a drug, and pharmacodynamics is what a drug does to a body. So these are the main endpoints. These are the main things that I work on. So there in pharmacodynamics we have viral loads, etc. And in pharmacokinetics we have AUC, area under curve, and then Cmax, which is the concentration at maximum, Tmax, half life, and half-life of a drug. These are some of the endpoints that we work with. And these are the study designs. Mostly, this is a parallel design or a crossover trial. And so we already know that how a parallel design and a crossover trial works for. So one of the project that I worked on was a CQTC analysis. It was a single in molnupiravir. It was a single ascending and multiple ascending dose study, which we combined together other dose levels. And the main target for a CQTC analysis is like torsity points is a disturbance of heart rhythm, and we want to see if torsity points is associated with QTC prolongation. Uh, it is associated with QTC prolongation and leading to heart life-threatening diseases and sudden cardiac death. So our main target was to see whether this drug leads to QTC prolongation, and we did some modeling and mixed effect modeling, and looked into the data and did some safety analysis, and then we see that this drug doesn't prolong CQTC analysis. And this is a scientific white paper that is being used all over the industry to um, do this type of CQTC analysis. So this is the drug that I worked on molnupiravir, and this on the left and on the right hand side of mine, this one, this is one of the paper cuttings, newspaper cuttings from India, uh, that my mom sent me, and it is written in language Bengali. And so this it was a clinical trial conducted in hospital in Kolkata, India. So it's a Bengali newspaper, and we started like it is very close to my heart that I'm working on this kind of drugs, and working on CQTC analysis of this kind of drug. So this is pretty close to my heart because I uh, can see the direct impact of it to the population in India, back in India, and to my hometown. So I
Arijita, the next session is supposed to start at 9, which is now.
Okay, sorry about that. Let's skip. Okay, thank you so much for your time. Really appreciate all the audience attending it. Thank you.